I'm Drew Chong. I'm the head of target discovery at Hydra Biosciences, and we're a biotech startup in Cambridge, Mass. We have about 20 people, and the real focus of our company is discovering novel agonists and antagonists of ion channels.
What did you do after your undergrad?
So after my undergrad, I ended up going to graduate school at Stony Brook in New York, and I did my PhD in the Department of Neurobiology and Behavior there, really studying the genetic regulation of ion channel expression and function in mammalian cells. And then from there I went to Harvard and went to Children's Hospital, where I did a postdoc in a lab there. And my advisor there started Hydra Biosciences and asked if I wanted to come on board as a scientist here. And so, although my degrees, undergraduate degrees are just in general biology, and my graduate degrees in neuroscience, I've spent a lot of the last 15 years working in drug discovery. And so just applying the techniques to a new system.
And then at Hydra, what are you guys trying to accomplish?
So we're trying to develop novel medicines for unmet medical need. And we're really focused on a group of ion channels called the TRP channels. These are a relatively newly discovered set of ion channels. And ion channels are really proteinaceous holes in the cell membrane that allow communication between the inside and the outside of the cell. And it's ion channels which are the main drivers in the nervous system. The nervous system is an electrical system, and instead of a copper wire you have axons, and instead of electrons you have ions flowing into and out of the cell. And so the signaling in the nervous system is done through the ion channels, and so we feel that that's a point where we can attack diseases which haven't been well attacked before.
Although this is mainly like a biology-based company, how do the chemists play a role in the company?
So for us, we use chemistry in order to design and synthesize novel inhibitors of the ion channels that we're interested in. A typical project at Hydra starts with an idea, something like inhibiting this particular ion channel would be good for treating this particular disease. And the first step then is to clone that ion channel and express it in a cell line where we can measure the activity. And so, we do a lot of molecular biology here and cloning of the ion channels, and then a lot of cell biology where we transfect the cloned channel into a cell line where the activity of the channel can then be monitored either with fluorescent dyes or monitored directly electrically. And once we have the channels cloned into the cell lines and we have a way to monitor their activity, the next step is to screen a library of compounds in a high throughput manner in order to determine which of the compounds in the library inhibit the channel. So a typical high throughput screening campaign is somewhere around 100,000 molecules. And this is representative of a much larger library of potential molecules yet to be synthesized or that are available from other sources. And so the idea initially is to screen through all those 100,000 compounds that we have already synthesized in our library, and then a medicinal chemist look at the compounds that are inhibitors and try to understand just a priori from looking at the molecules, what about the molecules makes them inhibitors of the channel, and how many different classes of inhibitors might be represented in the library. And before I started doing drug discovery, I thought that the high throughput screening was kind of the end of the drug discovery process, and that one of those molecules that you screen would then be the drug that you move forward. And it turns out that really high throughput screening is the beginning of the drug discovery process, and it's the medicinal chemistry that goes from taking the initial hits and then inventing the drug based on that data that comes from those hits. So it turns out that in order to be a good drug, you don't have to just a particular molecule doesn't have to just inhibit the target. It has to inhibit the target in a selective way. And then beyond being selective, it also has to be very potent, and it also needs to be non-toxic, and it also needs to be bioavailable in humans. And also has to have a reasonable, let's say, half-life in humans, so that you're not having to take the drug every 5 minutes, but that maybe 1 or 3 times a day dosing is going to be sufficient to be inhibitory of the target over time. And so the high throughput screening library is synthesized on a relatively small scale, and once we have hits, and those hits are being resynthesized by the chemists, and also they're being functionalized in different ways and being expanded upon to make new molecules that could also be inhibitors. We have to go back and test those again. And so the medicinal chemistry process is very iterative between biology and chemistry, where the chemists are constantly making new molecules to be tested, and then the activity of those molecules is measured, and that data is fed back to the chemists who then design new molecules. And it's a kind of a multi-parameter optimization process where you're trying to optimize, like I said, for both potency and selectivity and toxicity and bioavailability and stability. And all of these parameters are being tried to optimize at the same time. So it's like trying to keep 5 balls in the air at the same time, because you can drive down potency, but if you lose selectivity at the same time, you're no closer to being a drug. And so typically we synthesize molecules on a relatively small scale, about 5 milligram scale initially. And to do this, the chemists will do standard chemistry reactions, but when we're in the initial hit expansion phase and we're trying to find lots of compounds that are related in some way to the hits, and we don't know exactly what molecules are going to be, or what areas of the molecule are available or amenable to modification, what they'll do combinatorial chemistry. And the idea there is that you take a single molecule and functionalize it, the core of the molecule, and then you can also have lots of parts that would stick to the outside of the core that are also functionalized, and you can swap them in and out, a bit like Legos, like taking different colored Legos from the bin. So you have one piece that's functionalized, let's say for an amine reaction, and then you'll have all the other bits functionalized in the same way, and you can click them on one at a time and then purify those molecules and then test them for their activity. So it's more of the biology lab where we do a lot of the cell biology, which occurs in here, where they do typical tissue culture in the hoods, and then there's more molecular biology. There's the high throughput screening area in here where we monitor the activity of the channel in the cells in a high throughput way. And then we have the chemistry. The typical chemistry lab where they're doing, lots of different reactions, standard organic chemistry, refluxing, purification. It's just the combi flash where you can put in multiple different molecules and react them to form new molecules and then purify them automatically. We have an LCMS, which allows us to determine the molecular weight of the molecules that we've synthesized and confirm at least that they have the correct molecular weight. And so it's one of the ways to determine whether the molecule that we've synthesized is actually the molecule that we were trying to synthesize. A typical reaction that our chemists are doing here is being done in a fume hood, so that any noxious fumes that are generated during the synthesis are being vented instead of coming out to the air inside the lab. And also the plastic or the glass doors are there in case there's an explosion that's contained inside the cabinet or a fire. And this particular reaction is being done under hydrogen. So in order for the chemical reaction to occur appropriately, hydrogen, it's being done under an atmosphere of hydrogen, and it's being not heated at all, but it's being stirred on this. And so the reaction is occurring during the agitation process. So one quick and easy way to determine if you've made the product that you think you have is by thin layer chromatography. And so here are some samples that have been run in thin layer chromatography. The samples were spotted, and then you can see the product that is run up, and that the distance of the run compared to the standard should be characteristic for a particular molecule. The melting point is characteristic for a molecule as well, and so another quick test you can do is to determine the melting point on something like this, and it's just a visual system where you put a little of the crystal in there and visualize it as it's heating up, and then when you see that the crystal has melted, you note the temperature. So although Hydra is not a chemistry company per se, we're a biotech company, chemistry really plays an integral role in everything we do, and is medicinal and synthetic chemistry is going to be as important to our success as the biology underlying the diseases that we're targeting.